What if the story of evolution isn't the full story? What if human beings were altered, not by nature, but by design? Geneticists are uncovering patterns in our DNA that look less like biology and more like software. In this episode, we'll explore 6 disturbing clues that suggest humanity was engineered, not evolved, by a higher intelligence. There's a single gene that might explain why we became human. It's called ARHGAP11B, and it's not found in chimps, gorillas, or even Neanderthals. It exists only in modern humans. What does it do? It causes rapid expansion of the neocortex, the region of the brain responsible for language, abstract thought, and consciousness. In other words, the things that make us, us. But here's the problem. ARHGAP11B appears suddenly, not as a slow mutation, not as a gradual shift over generations, just there, as if it was added manually. According to Dr. Marta Florio, a neuroscientist at the Max Planck Institute who studies the evolution of the human brain, the gene wasn't inherited. It looks like it was spliced from a longer, inactive segment and reassembled into a functional code. In case you are wondering, that's not how evolution works. Natural selection doesn't rearrange and rewire dormant sequences with surgical precision. It doesn't build entirely new brain-expanding genes out of nowhere. Yet here we are. Without ARHGAP11B, we wouldn't have cities, language, or civilization. With it, we became something else. So the question is, who or what inserted the code? Humans have 46 chromosomes, but all of our closest relatives, chimpanzees, bonobos, gorillas, have 48. Why? Because at some point, 2 ancestral chromosomes fused into one. This became human chromosome 2. That kind of fusion is rare and dangerous. When 2 chromosomes accidentally combine, it often results in miscarriage, sterility, or severe developmental disorders. Yet for humans, it worked perfectly. Even more bizarre, the fusion point is clean and traceable. Scientists can literally see the 2 centromeres and telomere remnants. It's like an old weld scar in our genome. But that's not the strange part. Right where the fusion occurred, scientists found something else. A concurrent genetic mutation that activates a critical gene called PRDM16, which plays a role in placental development and embryo viability. In simpler terms, the fusion, which should have prevented reproduction, came packaged with a fix that made pregnancies succeed. The odds of that happening randomly? Astronomical. It's as if the genome knew the fusion was risky and preloaded the patch. Nature doesn't usually work this cleanly, but engineers do. There's a region in our genome called HAR1, short for Human Accelerated Region 1. It's nearly identical across all vertebrates: turtles, chickens, chimps, you get the idea. But in humans, there are 18 sudden changes. No slow transition, no fossil trail of edits, just a jump. And HAR1 isn't random. It's active in one very specific place: the developing human brain. Scientists call this a mystery jump, a sudden burst of precision mutation. But precision doesn't look like mutation. It looks like a software update. That update caught the attention of evolutionary geneticist Dr. Katie Pollard, who published a groundbreaking study in Nature in 2006. She and her team found that HAR1 had remained virtually unchanged in most animals for hundreds of millions of years. Then suddenly, And only in humans, it evolved at a rate 18 times faster than normal. To Pollard, it made no sense. We were shocked by how rapidly it changed, she said. It was like someone had hit fast forward on one of the most stable regions in the genome. The changes occurred not just anywhere, but in embryonic brain tissue, right where the neocortex is forming. And the neocortex is where abstract thought, memory, and consciousness take shape. Why would such a hyper-conserved sequence mutate only in humans, and so precisely in the part of the brain that separates us from every other species? Unless it wasn't random mutation. It was a targeted modification, an edit, a patch, a nudge from something that knew exactly what it was doing. In 2013, two researchers, Maxim Makakov and Vladimir Sherbak, published a peer-reviewed paper proposing a bold idea: that human DNA may contain an embedded message, planted by an advanced intelligence. Not metaphorically, literally. They described finding a set of arithmetical and ideographical patterns in our genetic code, structures that followed symbolic logic, not biological necessity. For example, when you classify codons by atomic mass and decimal symmetry, they form perfect numerical alignments, like multiples of 37, and palindromic structures that follow precise formatting rules. Nature doesn't create base 10 symmetry. Software engineers do. Makakov and Sherbak called it biological SETI, a genetic version of the wow signal. A transmission hidden not in the stars, but in us. So, what exactly are palindromic structures in DNA? Well, they're sequences that read the same forwards and backwards, like the word level. But in genetic terms, they're more than just symmetrical. These mirrored patterns often act as insertion points in the genome, precise locations where genetic material can be added or edited. In modern labs, we use these same palindromic regions with tools like CRISPR, a revolutionary gene-editing technology that allows us to cut and rewrite DNA. So when we find these mirrored sites, arranged with, let me remind you, mathematical precision, already embedded in human DNA, it raises a disturbing question. Who was editing us before humans even knew what DNA was? If Makakov and Sherbak are right, we're not just looking at raw biology. We're looking at evidence of intention. A message sent not through sound waves or satellites, but written directly into bloodlines. Across human populations, we see dramatic differences in average IQ, immune response, and even behavior. This isn't about superiority, it's about variation that appears too structured, too directional to be random. Some theorists suggest, what if different versions of humans were made for different purposes? This matches what we see in ancient civilizations, like the Indian caste system or Sumerian social hierarchies, where certain people were born to rule, others to serve. Could this reflect a genetic stratification, seeded not by nature, but by an intelligent hand? And if so, who determined the roles? Sometimes, the oldest records are the most revealing. In Mesopotamian tablets like the Atrahasis and Enuma Elish, humanity wasn't made out of love, but out of necessity. The gods, tired of laboring, commanded the deity Enki to create a replacement species. That species was us. The Atrahasis even describes the process, mixing clay with the blood of a slain god, a recipe that eerily resembles genetic engineering. This idea surfaces again in the Bible. Let us make man in our image, after our likeness. But who is us? And why say likeness? unless something is being copied. Then there's Genesis 2:5, often ignored. There was no man to till the ground. In other words, humans were created because there was work to be done. We were made to serve. Across cultures and millennia, the story stays eerily consistent. We weren't made to thrive. We were made to work. So the question lingers, who made us? And are they still watching? So, what does it all mean? Why are there fingerprints of design in our brain, our genes, and our oldest myths? Are we the result of a billion-year accident, or the product of someone else's blueprint? Were we bred, herded, and sometimes culled like livestock in a cosmic pasture? And if so, who are they? What do they want? And are they living among us still? We'll explore these questions and more. Watch the skies. Watch the patterns. Watch Nordic Files.